Thank you. My name is David Steed. On behalf of Noveome Biotherapeutics, I wish to thank the OIS for the opportunity to present our work. Our forward-looking statement. ST266 is our one-of-a-kind product. It's a unique cell-free platform biologic with powerful healing capabilities in treating multiple diseases and injuries given by different routes. It's made by a scalable manufacturing process for all indications in our pipeline. In traditional drug development, one drug works on one target. For example, penicillin, an antibiotic, affects one pathway to inhibit bacterial growth. ST266, on the other hand, contains hundreds of biological molecules, mostly proteins, in physiologic amounts, picograms and nanograms, acting in concert on hundreds of targets to control a disease or injury. Physiologic concentrations improve safety by reducing the likelihood of drug-related adverse events using such small picogram and nanogram doses. Single drugs are commonly given in amounts 1,000 times greater. This slide represents the production process for ST266. The components of ST266 are secreted by a novel population of cells generated by a proprietary method of culturing amnion-derived epithelial cells collected from full-term placentas normally discarded after birth. These cells are tested, released for production, pooled, then cultured in a bioreactor. The cells are later discarded, and the cell-free product is ST266. The multi-targeted secretome contains growth factors and cytokines which act synergistically to stimulate a variety of anti-inflammatory and neuroprotective pathways, resuscitate damaged cells, and thereby enable restoration of the natural healing process. ST266 offers the ability to improve outcomes in complex diseases for which single-target drugs are inadequate, or delivery of the drug proves to be an insurmountable challenge, such as crossing the blood-brain barrier. A scalable manufacturing process is used to produce ST266 drug substance for all indications. A drug master file has been submitted to the FDA supporting all IND applications. Noveome is focused on using ST266 given by way of intranasal transcribiform delivery, thereby bypassing the blood-brain barrier. We are developing a pipeline of products with broad therapeutic potential by multiple mechanisms of action in ophthalmology indications. Noveome is currently open to enrollment in a phase 1 dose-ranging safety trial of ST266 given to glaucoma suspects with mildly elevated intraocular pressure but no glaucomatous changes. This safety trial will serve as a prelude to a phase 2 trial in glaucoma and also one in optic neuritis. This is a schematic for ST266 given to the optic nerve by intranasal delivery. Preclinical studies are completed, the phase 1 trial is underway, and the phase 2 trials in glaucoma and optic neuritis are planning. This illustrates the method of intranasal transcribiform delivery. This is not the same as most nasal sprays. This drawing is a sagittal plane through the left nostril. The delivery device directs the plume to the cribiform plate, which has foramina through which the olfactory nerves make their way to the nasal passage. These same foramina carry ST266 to the brain, bypassing the blood-brain barrier. Here are data from a mouse model of optic nerve disease using the experimental autoimmune encephalomyelitis antigen, EAE, which mimics multiple sclerosis. On the x-axis is the time in days, the y-axis is visual acuity. At the top, the green line is control animals who did not get EAE and had no change in visual acuity. The light blue line at the bottom is animals who received EAE and no other treatment. Their vision worsened and they were blinded at 6 weeks. The red line here is animals who received EAE, their vision worsened. They were then treated with ST266 by intranasal transcribiform delivery starting at the end of week 2. Their vision returned to near normal by 6 weeks. The dark blue line is also animals who received EAE. Their vision also worsened. They were treated with ST266 starting at the end of week 3. They then had returned to their vision to near normal by the end of 6 weeks. ST266 clearly reversed the visual impairment caused by EAE. This experiment was repeated with the same outcome. These are the results of an acute mouse model of optic nerve trauma. The x-axis is time in days. The y-axis, again, is visual acuity. The optic nerve was crushed with a clamp. The green top line is control animals who did not undergo optic nerve crush. The light blue line is control animals who had optic nerve crush but were not treated with ST266. They were blinded. The red line is animals who received ST266 by intranasal transcribiform delivery after the optic nerve crush. These animals had a statistically significant preservation of about half of their vision. Thus, ST266 salvaged vision in both acute and chronic mouse models. This is our timeline for our phase 1 safety trial, now open for enrollment. It should be completed by the end of the first quarter of 2020 and data analysis by the end of the second quarter of 2020. In summary, Noveome has developed a patented platform technology containing multiple cytokines used for multiple indications, delivered by multiple methods. We've established a scalable GMP manufacturing process. We've demonstrated safety in 237 patients in 9 trials and efficacy in 3 trials. We are currently enrolling patients into the phase 1 safety trial of intraocular hypertension, that is glaucoma suspects, using transcribiform delivery of ST266, and are planning a phase 2 safety and efficacy trial in optic neuritis and glaucoma. Noveome thanks the Ophthalmology Innovation Summit for the opportunity to present our work.